And so within your brain are tiny molecular messenger switches that control how it develops, learns, and even how it reacts to stress. One of these switches involved is a process called acetylation, specifically H2 acetylation, which acts like a light switch turning on genes essential for function. During prenatal development, when a mother experiences stress, her body releases stress hormones like corticosteroid. These hormones can influence the developing brain, especially in regions like the hippocampus, which is crucial for memory and spatial navigation. Elevated stress might lead to changes in DNA methylation, another molecular process where methyl groups are added to DNA, often silencing gene expression. Enzymes called DNA methyltransferases are the craftsmen behind this, adding methyl groups to specific DNA sites. These epigenetic modifications like methylation and histone modifications such as phosphorylation and acetylation are like decorators changing the brain's blueprint. They influence genes like linked to memory, learning, and even behavior. Disruption in these processes can lead to developmental delays, motor coordination issues, or neurological conditions like epilepsy and breathing abnormalities. Some individuals may also display autistic behaviors, language impairments, or work memory deficits. These outcomes can be linked to haploinsufficiency, a situation where one copy of a gene isn't enough for normal function. Within the neural landscape, transcription factors like helix loop helix proteins bind to DNA and regulate gene transcriptions, influencing neurodevelopment and function. Post-injury, such as traumatic brain injury, these epigenetic mechanisms come into play, either helping repair or contributing to cognitive dysfunction. An enriched environment can promote beneficial epigenetic changes. This includes enhancing cholinergic signaling, which involves the neurotransmitter acetylcholine, vital for attention and memory. The nucleus of these processes often involves nuclear receptors and signaling pathways that activate a CAMP response element binding protein, PrEP, a key player in turning on genes necessary for learning. On the surface, receptors like NMDA and ionotropic glutamate receptors are gatekeepers of calcium ions entering neurons, initiating signaling cascades that lead to gene transcription. These pathways activate proteins like neuronal pentraxin and neurotin, which support synaptic growth and plasticity. Cannabinoid receptors and apolipoprotein E modulate neural communication and repair. Small interfering RNA to selectively turn off problematic genes is what scientists use to provide insights into how gene expression shapes brain development. Enzymes such as histone deacetylases can remove acetyl groups from histones, tightening DNA winding and silencing genes where histone acetyltransferases loosen that tight grip. So next time you think about your brain, remember it's not just a static organ, a dynamic, ever-changing state with molecular switch and pathway constantly working behind the scenes to keep you sharp, adaptable, and resilient.